Hello class, this is a post-lab discussion on alcohol. We recall primary, secondary, and tertiary alcohol. Okay, so this presentation will cover iodoform test, oxidation condition, rate of oxidation, and comparison of rate of substitution. For iodoform test, the procedure consists of adding sodium hydroxide and iodine in Ki into the test solution. Secondary alcohol with methyl group on the carbon carrying the OH, such as in isopropyl or propan-2-ol. Methyl group bonded to the carbon carrying the hydroxyl group, or compounds like this will give a positive result. Ethanol is the only primary alcohol which will give the iodoform test reaction, and ethanol the only aldehyde. Iodoform can also test for ketone and secondary alcohol. Okay, so this is the chemistry involved. This is ethyl alcohol, sodium hydroxide, and iodine in Ki to form, this is the yellow insoluble product. Okay, likewise dito, this is the iodoform, this is the iodoform. Okay, so the test result, this is a negative result. This is the result for 1 propanol, 1 propanol. This is the result for 2 propanol and for acetone. Permanganate oxidation. Permanganate ion, or MnO4 -1, oxidizes both primary and secondary alcohols in either basic or acidic solution. Okay, so the reaction is going to be faster in basic or acidic condition. It can also take place in neutral condition, but it's going to take place very, very slowly. With primary alcohol, the product is carboxylic acid because the intermediate aldehyde is rapidly oxidized to carboxylic acid. This is the uh, chemistry involved. In acidic solution, this is what is produced from purple permanganate to Mn+2 or manganese 2 ion, which is colorless. In basic solution, this is a chemistry involved formation of manganate ion, which is green. And in neutral condition, in neutral condition, the reaction takes place very slowly. What you produce is manganese dioxide, which is brown. In the slide, you are shown the test result when it is acidic, from purple to colorless, and when it is neutral, from purple to brown product. The precipitate here is the manganese dioxide precipitate, which is brown. Dichromate oxidation. Okay, so condition, potassium dichromate in concentrated sulfuric acid. Primary alcohols are oxidized to aldehyde, and it will stop at the aldehyde stage. Hindi na siya mag-proceed to carboxylic acid. Okay, so the chemical reaction from yellow color of the dichromate to blue-green. Okay, the test result, um, for ethanol, a little green color. For phenol, for ethanoic acid, what is ethanoic acid? It's acetic acid. And for 2-hydroxybenzoic acid, these are negative results. These are positive results. okay, with propan 1-ol or 1-propanol. Green, okay, green also for propan 2-ol or isopropyl. And this is the negative result. This is the principle involved in alcohol test, breath test. If it tells if you are drinking while driving, there is going to be a color reaction. And the Lucas test, the difference in reactivity of primary, secondary, and tertiary alcohol is demonstrated in Lucas test. In this method, the alcohol is treated with Lucas reagent, which is a mixture of concentrated hydrochloric acid and anhydrous zinc chloride. The alcohol is converted to alkyl halide. Alcohols are soluble in Lucas reagent, while the halides are insoluble. The formation of alkyl halide is indicated by the appearance of turbidity in the mixture. As the reactivity of alcohol with halogen uh, is in the order of tertiary, is faster than secondary and faster than primary. The time required for the appearance of turbidity will be different for primary, secondary, and tertiary alcohol, which helps to distinguish them from one another. Okay, so this is the chemistry involved. This is a substitution reaction. The hydrogen halide, okay, the halogen replaces the OH group. So you will form the alkyl halide. Okay, so this is the test reaction. This is a negative result for primary alcohol, a turbidity for secondary alcohol, and separation of layer for tertiary alcohol. Okay, so this is the condition uh, if this is the test result if the condition is carried out room temperature, walang heating. So only at room temperature. Okay.